society and the public conversation is grappling with a whole new era of genetic modification. And what am I talking about here? I'm talking about the CRISPR technology. And for people that don't understand what this is, I'm going to show a little animation. The CRISPR technology is basically the editing of the genome. So what the scientists do is they can clip certain parts of this genome with these genomic clipping scissors, and they can take pieces out, they can put pieces in, it recombines, and that really makes a whole new genomic species at that point. Where the old GMOs, if you will, the GMO foods that we talked about, they were inserting genes. They were inserting herbicide-resistant genes, insect-resistant genes.
Right.
We're not inserting anything. We're going in and clipping and splicing and doing what we're told is targeted things with
almost
this.
like surgery on your DNA or something, right?
Yeah.
And then this idea, I mean, it's been, for many years now, we keep waiting for CRISPR technology to come out and save us from cancer and all these things,
right.
But it just seems to be stalled.
Right, and that's what we were sold on. This is one of the big things, is it's going to help people. You just clip out those inconvenient parts of your genome that give you chronic illnesses and diseases and cancers. And so what we started to see, some of the first headlines in 2018, 2020 were headlines that looked like this. There were 2 studies that came out. CRISPR-edited cells linked to cancer risk in 2 studies. So they're finding their CRISPR editing is actually leading to a genomic change that is causing cancer. So that's what some of the studies are finding. So let's pump the brakes here for a second. And so we start to look into this phenomenon in the human capacity, in the human cells. And we have a study here. This was in 2022, and they were looking at on-targets. They were looking at specific parts of the organs that they would actually use, these on-target cells, to fight these diseases with CRISPR. And this is what the researchers found. Let's check out this study.
Yeah.
So they said, they used a word called chromothripsis. Now remember that word, as an on-target consequence of CRISPR-Cas9 genomic editing. The Cas9 is kind of like the newest guy in the block. It's the fastest, cheapest way to do this. It's the newest technique. It's the most accurate technique, we're being told. And so what did these researchers find when they looked at these on-target human cells? This is what they reported. They said, here, using model cells in single-cell whole genome sequencing, as well as by editing at clinically relevant locus and clinically relevant cells, we show that CRISPR-Cas9 editing generates structural defects of the nucleus, micronuclei, chromosomal bridges, which initiate a mutational process called chromothripsis. Chromothripsis is extensive chromosomal rearrangement restricted to one or a few chromosomes that can cause human congenital diseases and cancer. Now, this chromothripsis is this instantly cascading effect of these rapid changes. The scientists, the researchers, geneticists, they don't know what's going to happen at the end. They don't really know what it does downstream, but they know it's happening. They can witness it in the lab, in the cells here.
Wow.
It's very, this is a phenomenon that us as the public and some scientists are saying, wait a minute, is anybody looking what that's doing? And they're saying, no, we got CRISPR-Cas9. This thing is so accurate. We can just clip these cells and look what we're doing. And now we get to a new study that just came out, and it looked at the CRISPR technology in a tomato. So now we're getting directly to our food shelves, directly to our dinner tables. And this is from GM Watch. They've been really a watchdog in the genetically modified space.
Yeah.
Headline here, gene editing found to cause chaos in the genome of tomatoes. And they look at this study. They're calling it CRISPR-induced DNA break can trigger crossover chromosomal loss and chromothripsis-like rearrangements. We have an independent impact assessment test biotech organization, and they're actually calling it CRISPR-thripsis because it's such a normal phenomenon. They're calling it CRISPR-thripsis in plants. And they put out this warning after that study came out. They said the recent findings shed new light on the alleged precision of gene scissors. Although the new technology can be used to target and cut precise locations in the genome, the consequences of this, quote, cutting the genome are to some extent unpredictable and uncontrollable. Plants obtained from this new genetic form of engineering cannot, therefore, be regulated as safe per se and need to be thoroughly investigated for risks. And this is where we're at.
It's exactly, and we sort of, because we're here at Freedom Fest, we thought, let's just not bury this in all the vaccine work we've done. But this is exactly the problem. It's why you're watching David Spade and Dana Carvey have to crack jokes about how ineffective the vaccine is, because the science was done this way. We made a vaccine out of mRNA, had never been done before, and all we look to see is, did it make antibodies? It made antibodies, great, put it on the market.
Yeah.
Forget about how long do those antibodies last? Don't know.
Right.
Do those antibodies actually, create any sort of protection? Don't know. Right. Are there any other health outcomes that could be be being created by having these antibodies in your body, or a spike protein floating around, a mutated spike protein? Don't know, didn't look at it, didn't do any long-term trials, and now people are, dealing with the consequences worldwide of decisions like this. And I always say to people that challenge me, they're like, oh, the COVID vaccine is perfectly safe. I said, well, if you go online, you'll find that probably isn't true. But even if it was a success, do we really want to live in a world where you're going to rush products out that have never been injected into human beings, never really worked in a piece of fruit or a vegetable we're about to eat? Do you want these products rushed out within 12 months so that you have no idea what happens to your body 2 years down the road, 3 years down the road, 10 years down the road? What about your offspring? Is it affecting the DNA that you're passing on to your offspring? Do you really want to live in a world where the pharmaceutical industries and these big agrochemical companies are able to change your food, change your body with no understanding of the long-term effects, and they can crank it out in 12 months because we're in a rush?
Right.
We got to do this because we just told your government that it's an emergency to make this happen right away. I mean, this is... If these technologies haven't already destroyed our species or sending us down that road, if we stay in here, if we keep letting products being made this way, there's no way we survive this.
Right.
We will eventually make a scientific mistake, I believe, that will wipe our species off the planet. Certainly, when you think the entire reason these products are being made this way is to somehow beat our war with nature, nature has never wiped our species off the planet in all the time that we've been here as human beings. But we keep making mistakes like this, I think we'll be the ones that do it to ourselves.